Hey everyone, in this video lecture, we will be talking all about mitosis. Before we can talk about mitosis, we need to review the steps of the cell cycle. So, the cell cycle is a series of steps that a cell will go through before it's getting ready to divide. And this is particular only to somatic cells or cells that make up your body. So there are three main tasks. Task number one is to have that cell get bigger, larger in size as it's copying all the contents of the cell. Then there's also a step where the genetic material, so the DNA, needs to be copied as well. And then lastly, the task we're going to talk about today is that one cell will split into two new cells. So, mitosis is that step or a series of steps where one single somatic cell will divide into two identical somatic cells. And the key is identical. The organelles, the genetic material, all needs to be exactly the same at the end of mitosis. So, we're going to see the steps involved for how we get two identical cells. There are four phases for which mitosis occurs. The acronym to remember the order of these phases is PMAT. The first phase is prophase, followed by metaphase, anaphase, and telophase. So we'll go through what is occurring within the cell at each of these steps in order to make these two identical cells. So the first step, prophase, this is where that cell is really truly beginning that step of dividing. So, one important mechanism for helping a cell divide is these things called spindle fibers. So, at this step, they begin to form out of your centrioles, which is the organelle that helps cells divide. Spindle fibers, think of them like the pulleys of a cell. They're going to pull apart the organelles in order to actually separate and make two separate cells. The nucleus or the nuclear envelope that surrounds the genetic material is going to start to break apart because we need those chromosomes inside of that nucleus to be able to move around the cell in order to divide. So the nucleus is going to start to break away. At this point, your DNA is wound up into nice, pretty packages called chromosomes. Okay, so DNA is usually in this loose kind of ball of yarn state, if you will. So, in order to divide the DNA properly, DNA winds up on itself to make a nice, neat package called a chromosome. And this makes your sister chromatids visible. And we're going to talk about what a chromatid versus a chromosome is. So, a chromosome is again one package of DNA. So, depending on how much DNA an organism has, there might be more than one chromosome. And what happens is that chromosome, that set of DNA, is going to replicate during the cell cycle. So, after DNA replication, there are two copies of that chromosome. So, to distinguish whether it's one copy of one chromosome or two copies of one chromosome, we use the terminology of sister chromatids. I really think twin would have been a better term here than sister, but sisters to tell you that they're identical, they're copies of each other that are connected together. So this is still one single chromosome. It's all the same genes, all the same part of DNA. However, there are two identical copies. So we call each copy a chromatid. This is definitely very confusing terminology to make sure that you understand. So chromosome is one single part of DNA wound up tightly in a nice pretty package. But after DNA replication, there are now two of those packages, but they're still the same DNA, so they're the same chromosome, but each copy we call a chromatid. So I know it's confusing, so you just really need to practice with the vocabulary. All right, so now that we have those chromosomes visible, during metaphase, what's going to happen? Metaphase begins with the letter M. We want to think of the word middle. because what's going to happen is the spindle fibers, again, think of them like the pulley system of the cell. They're going to attach to your chromosomes and start to move them. And they're going to move them to the middle of the cell. So during metaphase, those chromosomes are lined up in pretty much a nice straight line in the middle of the cell. Then anaphase begins with the letter A. We want to think of the word away because those sister chromatids, again, they're the identical copies of the same DNA, are going to start pulling away to opposite ends of the cell. So those spindle fibers are attached to the chromosomes, or the chromatids, I should say, and they're pulling them away to the opposite ends of the cell. Then the last part of mitosis is called telophase. And so at this point, the cell is really kind of done separating. So those spindle fibers are not needed. They are breaking apart. They're just kind of dissolving away, if you will. And those chromosomes are now where they need to be. So they start to decondense. So the DNA will start to unwind and, disassemble from that nice wound up package of DNA. And the way to remember the biggest part of telophase begins with the letter T. And at this step, two nuclei are beginning to form. So now we can kind of start to see two distinct cells. Each set of DNA is being enclosed by a nucleus. So that is the end of mitosis, but we still actually haven't separated two separate cells. So that's the next step after mitosis is cytokinesis. So this is the last part of the cell cycle where we have actually divided up the cytoplasm to create two separate identical cells. So think cyto is cytoplasm. The cytoplasm divides to make your two separate cells. So if you need a little review, I highly recommend watching this Amoeba Sisters video as it does a good job summarizing the steps of mitosis.